Okay, thank you. So my name's Tim Millar. I'm from the New Zealand Institute for Plant and Food Research, and I'm going to give a couple of talks today. This first one is a bit of a mouthful of a title, so I apologize for that. But Jeff gave a really actually nice kicking off point for this talk. So this is a talk about this paper. Well, not really about this paper, about implementations of this paper that have been produced. So this was a paper from VanRaden in 2008. This is a really important paper, been cited many times. It's from the cattle literature around genomic prediction and particularly relationship estimators for genomic prediction. Now, this paper introduced, I believe, was 3 different estimators for genomic relationships. I'm going to talk about method 1 in here, which is probably one of the most widely used estimators for relatedness. And a really key detail in here that Jeff alluded to just before is that this paper is quite explicit that the allele frequencies used in method 1 should be from the unselected base population, so from the ancestral population, rather than from allele frequencies of the sample population after selection has occurred. And this is important detail because in practice, it's very common to use the sample population to estimate allele frequencies and use those in place of the base population. This is another example of that. This is Ashraf et al. 2016. This is an important paper in the polyploid literature because it was one of the first generalizations of VanRaden's method to autopolyploids. And in this paper, they are actually quite explicit that they use the sample population allele frequencies. And in their case, it seems like a very reasonable thing to do because they're working in a very diverse germplasm where you would expect the sample population allele frequencies to roughly reflect those of the ancestral population. But is that always the case? So at Plant and Food, I work primarily in kiwi fruit pedigrees, and these can become quite reticulate. So this is not a real pedigree, this is a simulated pedigree, but it has many of the features that we often see in some of our pedigrees. So we have a relatively constrained number of founders. We have quite a lot of reticulation and cross-generational crosses. And then at the bottom of that pedigree, we have a number of large terminal crosses, which are our large crosses for selection. And so here I'm just showing 20 advanced individuals, but you can imagine pedigree like this where you have a much wider base, where you have many more advanced individuals. So for the sake of simulating some data, I took that pedigree, simulated 10,000 loci evenly spread across 30 hypothetical chromosomes. Kiwi fruit has 29 chromosomes, and I decided to add one. I don't know why, I just did it at the time. So I simulated unique alleles for each of the founders. So these are effectively IBD alleles. Each founder has its own unique alleles. And then I simulated the inheritance of those genes throughout the tree via a gene drop simulation. Just note that this is a simulation without any recombination, so complete linkage. I played around with this a bit. You can also simulate with no linkage at all, so completely unlinked markers, and the results will of course change, but you'll get broadly the similar conclusions. So on the right-hand side, what you're seeing there is the true realized relationships as calculated from IBD against the pedigree relationships or the pedigree estimates on the y-axis. And you can see that that's the kind of result you would get. You have something that's fairly linear with the pedigree estimates, but there's some variation around that because of Mendelian inheritance. So taking that data, I then replaced those IBD alleles with biallelic markers. Now, in this particular case, I just replaced each of those unique founding alleles with a biallelic marker with the alternate allele drawn from a beta distribution. You can play around with the way that you assign those biallelic markers. It doesn't really change the results. And then I calculated VanRaden's first estimator via what I call the naive method, which is where you're using the sample population allele frequencies in place of the ancestral frequencies. So this is not actually what VanRaden described. I don't want to smear his name. This is not what he told people to do. This is just what's common practice. And you can see the fit we get there isn't great. So along the x-axis there, we have the true realized relationships. And on the y-axis, we have the estimator using the sample population allele frequencies. We get some pretty big outliers in there. And just to show that, so I did these calculations using a package called SGKit, which is in the Python side of things. But just to show that these are equivalent results from AGH matrix and SOMA. So in short, if you're calculating VanRaden method 1 and the function you're using does not explicitly ask for the base population allele frequencies, it's almost certainly using the sample population mean in its place because there's not much else it can be doing. So what if we do something better? This is the same calculation using the same package. However, now instead of using the sample population mean, I'm using just the mean allele frequencies of the founding individuals. We get something that's much more collinear. It's still offset slightly, and there are reasons for it. But importantly here, particularly for interpreting population structure, the rank order is relatively preserved. So the rank order of those relationships is quite consistent. Now that's all well and good, but often we're in this sort of situation where we don't actually have sequencing data or marker data for our founding individuals. So same pedigree as before, but now you can imagine we only have marker data for those individuals that are colored in black, and we have the pedigree record, but we don't actually have the marker data for those founding individuals. What can we do here? Well, if we apply the naive estimator again, then we get this result here, which is not terrible. Interestingly, those big outliers we saw on the first plot appear to be from the founding individuals rather than from the advanced samples. But there is something we can do to improve on this. This is a paper from 2004 that was in the diploid literature, but it's kind of, I haven't seen that many citations to it, but it's a really useful paper. So best linear unbiased allele frequency estimation in complex pedigrees. Sounds like a good fit. This equation is from not that paper, but a follow-up one from Harris and DiGiorgio in 2017. They apply this in mixed ploidy populations and polyploid, which is very useful. And I also find their notation a bit easier to work with, which is why I've used the equation here. So this is the description of the estimator. So the PI on the left-hand side there is the estimate of the founding allele frequencies, and the X on the far side is the individual allele frequencies for the sample population. And if we break this down, the K-1 there is the inverse of the kinship matrix as calculated from the pedigree structure. So essentially what this is telling us is that we get the sum of each column of the inverse kinship matrix and divide by the total sum of the elements in the inverse kinship matrix. For our users, that's weights equals col sums divided by sum of the whole matrix. And what this is giving us is effectively a series of weights for each individual in that we have sampling data for to calculate a weighted mean of allele frequencies. So those numbers are a bit small. This is the same pedigree as before, except now I've printed those weights on those sampled individuals. Along the bottom, you'll see that there's no weights at all, and that's because that estimator weighted all of those advanced individuals to 0. So essentially what this is telling us is that an individual with sample parents can't tell us anything about the ancestral frequencies that its parents aren't already telling us. So what you'll see when you calculate this is you'll get weights for basically the top of the sampled section of the pedigree. If we had samples for the whole pedigree, what you'd see is even weightings for each of the founders in that pedigree. It would bring us back to that founder estimate of allele frequencies. And if we calculate VanRaden using these blue allele frequencies, we can see that still not a perfect fit, but certainly an improvement in the rank order of relationships. So there's the comparison there. Only 5 minutes left. So just to give a try and intuitive explanation of the mechanism that's at play here, VanRaden's first estimator really emphasizes rare alleles based on those allele frequencies. And so if you have an allele that is very common in your founding population, but through selection and inbreeding has become rare in the advanced population, that means individuals that share that allele will assume to be closely related to one another. So if you have, for instance, a couple of founders that share what was a common allele, but it's treated as a rare allele by the estimator, it will make those founders look unrealistically related to one another. And that's what we saw with those really large outliers in the first plot. The inverse is also true. So if you have a rare allele in your founding population that has been concentrated by inbreeding, then individuals that share those alleles will have an underestimate of their relatedness. So in summary, VanRaden's first method is defined in terms of allele frequencies. It's common practice to use the sample population frequencies in place of the founder allele frequencies. This is reasonable in some populations, particularly if you have a very diverse population without much inbreeding. It becomes less reasonable as your population is more inbred, and also you get the severity of the effect increases with the size of those advanced crosses. So if I was to do that analysis with the exact same pedigree, but with more individuals in those advanced crosses, you just further the effect, and so you make it even more disproportionate. There are a couple of options here. One that we talked about is McPeak's method for estimating the ancestral allele frequencies. That's potentially a viable option. It's not always perfect. It does rely to some extent on having accurate pedigree information. And there is always a risk there that if you have genotyping error in one of those founders and it's highly weighted, then you'll make that error worse. There are other options. So one may be to take allele frequencies from unimproved germplasm, if you have that available to you, or to look at other estimators. And so I've been playing around with this estimator from Waring-Goodall in 2017. This is an estimator of something they refer to as beta in that paper, which is essentially kinship of the population scaled by the mean kinship of the population. But they do have an equation in there for trying to rescale that to something more appropriate to kinship. That's a really nice estimator for a couple of reasons. One is that it's already been generalized to autopolyploids, which was work by Timothy Bilton from Otago University, a fellow student there. And that estimator also generalizes nicely to mixed pluripopulations and multi-allelic markers. So it's a very, in my experience, a very robust and generalizable estimator, which is quite nice to play with. Right, a couple of acknowledgements. Bruce Ware and Marcus Davey, who both gave me feedback on this early versions of these slides. Good.
Any questions for Mr. Long? We got a couple minutes.
Yes, so nice talk. At the practical standpoint, have you seen like difference in terms of prediction ability?
I haven't looked into this in oh, sorry. So where the question is about how this has effects on prediction, I'm assuming you mean in genomic prediction. Yeah. So I haven't looked into that yet. My interest in this really started around understanding population structure, because I anecdotally heard comments that along the lines from various people that I don't trust my pedigree data because my G matrix tells me something different. And the question I have then as well, is it actually an issue with the pedigree matrix, or is it a question with the G matrix? And so I'm more interested in the population structure in this talk, but that's a very relevant question that maybe I can defer to Jeff on that one. I'm not sure if he can comment.
It doesn't affect prediction at all. That's, I think, I mean, I've been doing this a long time. I think it's the first time that I have encountered where I became worried about this was in the top at the coma. I mean, until you start looking at it, the inbreeding in particular, I think it's very the predictions that have any change on the result. It's the other.
You go, my guess.
Yeah. But Jeff Endelman says that it doesn't change the predictions.